Well, thanks very much. It's a pleasure to be here. Thanks very much for the organizers for inviting me here. I'm going to talk about a topic which is a little bit more formal. From time to time, I'm going to mention some theorems. But I'm going to try to make a presentation sort of on the intuitive level. So the topic is one of system identification in computational neuroscience. And I'm going to start with a motivating set of slides. I'm going to talk about olfaction in the fruit fly. I'm involved with this, so we're doing systems neuroscience. There's not much time to talk about it in details, but at least I'm going to give you a sense of why we are looking at the class of theoretical problems which I'm going to present here. So the basic premise is that we have a fruit fly here. We would like to understand how order waveforms are represented in the brain. And we are looking at the first two layers of neurons, olfactory sensory neurons and projection neurons. And on the left-hand side, you can see the fly from the top. There's a set of antennas there. Excuse me. Well, I'm not sure how this works. Okay. I should come here. So here are the antennae of the fruit fly. And the size here is something like 250 microns. The brain itself is maybe 500 microns. And there are roughly 1,250 olfactory sensory neurons on each side, on each antenna. And so what we're interested in is essentially... Okay, so what we're interested in is basically doing recordings of the olfactory sensory neurons, and we do this with some tungsten electrodes. So essentially, if we implant this electrode here very close to the soma of this neurons, we can get a near field potential, and then we can see spikes. And so the problem appears to be extremely simple. One applies some order waveform as shown here. This is a recording. This is all in vivo. And then one abstracts the set of spikes as being a raster where only the timing of the spikes is important. So the problem appears, in terms of system identification, appears very simple at the beginning. But as soon as we started this, we found out that there are some huge problems there. And the fundamental problem is that in system identification, you have to generate a waveform, and you have to repeat the experiment with the same waveform. Turns out that it's exceedingly difficult to do. And it took us a very long time. And here you see the first auto delivery system which works, which delivers an auto waveform, which is very precise as a function of time, so that when you repeat the experiment, exactly the same auto waveform is generated. This has created a lot of problems in the past. People thought that olfactory sensory neurons are very noisy. And the noise actually was due to the auto delivery and not to actually the spiking behavior of the neuron. So now we have essentially established a system where one can repeat the experiment. Here we are showing two trials. You see there's essentially no difference between the two auto waveforms. This is a function of time. This is roughly on the scale of seconds. And when we repeat the experiment, essentially the same waveform appears both in absolute values, but also in terms of percentage error. So we are working here around 1% error, something similar to what people are familiar with in analog electronics. So the best you can do is around 1%. Now, this... Sorry?
In the previous slides, two different variables.
Yeah, there are two different waveforms here. They are completely overlapped, so you cannot see the difference. But if you want to evaluate it, you can see that the difference is 1%. Okay, so the delivery is extraordinarily precise. The problem we have encountered now is, so we have a precise auto delivery system. Therefore, fundamentally, we can approach the problem of system identification of how the neuron encodes information. The problem is that the basic theory, which is available in the literature, is based on white noise analysis. So what you do is you essentially try to apply some sort of a white noise waveform as an auto waveform, and you want to make sure that when you repeat the experiments, you get the same thing. Then we looked at the spectrum, and the spectrum is not white. And no matter how you go about it, you just don't get white noise there. So the delivery is very precise, but as you can see, this is the 3 dB line. This is a drop of factor of 2. And so from the point of view of engineering, this is sort of okay in the sort of maybe 50 hertz bandwidth, but it's not white. And so as a result, we started looking at a methodology to do system identification without white noise. It's number one. Number two, repeating experiments in system neuroscience is really very time consuming. And so what we wanted to have is a methodology whereby we can identify the system just by using a single sample path, a single experiment. You don't want to repeat the experiment. So you don't want to compute the PSTH, if you like, in the language of system neuroscience. So this is what I'm going to talk about today. So now I'm going to start the more formal description. So we're assuming now that we have a model of a neuron, and it consists of a spike generation, capability, and some processing. We're assuming that the processing is linear, so we have a linear filter. And the spiking neural model can be anything: Hodgkin-Huxley, integrated fire with random thresholds, what have you. And so the question now is, identify the linear filter knowing the output and knowing the input. And I'm going to analyze the situation for 2 cases. One when U is a continuous waveform, so you're at the periphery, and one when you have actually a train of spikes, so you're somewhere upstream in the neural hierarchy. I'm going to assume here that from a systems neuroscience standpoint, this neuron can be identified. And so it's like the Hodgkin-Huxley setup. You can go in there, you can inject currents into the soma, you can identify the neuron in terms of spiking behavior. In other words, the axon hillock can be identified. However, dendritic processing is a different matter. It's very difficult to do, there's no access, etc. So we're going to assume that's unknown. And so throughout the talk, what I'm going to do essentially is this. I'm going to change the dimensionality here of the circuit. I'm going to have a filter or a bunch of filters. And here I'm going to essentially exchange, for different spiking models. I'm going to treat integrated fire in the beginning because it's easier to understand. And then I'm going to go to conductance-based models.
Where does the probabilistic, where does different trial here will look the same? It looks as if it's completely deterministic. So why different trials look the same?
Okay, so from our standpoint, this is a system which is mechanistic. So mechanistic, let's say, think in terms of Hodgkin-Huxley neuron. But Hodgkin-Huxley neuron might be driven, let's say the gating variables might be driven by white noise. And as a result, the circuit itself is intrinsically noisy. Okay? So the analysis I'm going to describe here is for the case where the neuron is not noisy in the beginning. It's easier to understand. But then we're going to then generalize it to the case where we have stochastic behavior there. Okay? And so if you like, this is a linear operator. This is a nonlinear operator. Okay? And it can be a stochastic nonlinear operator. Okay? So let me start first with the simplest model. So I'm dealing with the ideal integrated fire neuron. It's sort of the backbone of a lot of the analysis which I'm going to present here. It's easier to sort of grasp what's behind this. So the essence is already here. This is a nonlinear system. This is a linear system. Single filter at the input, integrated fire with threshold delta. And so essentially we're going to say that U and T are known, and we would like to say something about H. Now, there's a key result here. I'm going to present it intuitively. Okay? So essentially, the first result here, which is called conditional duality, what it says is the following, that in effect, as far as the filter at the input is concerned, the previous system and the system as shown here, the equivalent, meaning the I/O behavior is exactly identical. And now the question is, what is this box here? So you see in the previous slide, the box there was a filter H, so it was a simple operator, linear operator. And here we also have a linear operator, so it's a filter. But now this is the projection of the filter on the input space. So intuitively, the way you want to think about this is like this. The input space is some linear space. It's like a vector space, okay? And you have a filter here, okay? You explore this filter with a class of input signals. So a systems neuroscientist, what it does is has a class of input signals or stimuli, applies them to, let's say, the fly. And in this experimentation, it explores a part of the spectrum of H, okay? So as a result, you see, given the input space here, only P of H can be explored, not the whole H completely. So intuitively, the way you want to think about this, in particular if you are an experimentalist, if the filter has a bandwidth of 50 hertz, and your class of input signals is 25 hertz, you're not going to be able to know what's happening in the bandwidth between 25 and 50. It's just not there because the input signal does not explore that space. And as a result, all you can do is identify sort of a subset of H. And that is, we call the projection of the filter on the input space. This helps us a lot because now what we do is we swap the two. We construct here a filter whose impulse response is actually the input signal U, and then we put the projection of the original filter as being the input signal. And suddenly, the whole problem collapses in terms of analysis because this is now a classical neural encoding problem. It's not a system identification problem anymore. And the reason for this is that now U is known because we have complete control of it. The output is known because we record it. And then the question is, what is the input to the system? So this is classical encoding, like in information theory. And so you ask the question, knowing the box, meaning the transformation, and knowing the output, what can you say about the input? And this problem has a beautiful solution, which says that P of H can be computed with arbitrary precision. And so this is very now powerful from a theoretical standpoint because you know exactly how far you can go. But it also tells you that you cannot find out what H is. You can only find out what pH is. Okay? So that means that in experiments, if you repeat an experiment, you might get actually a different answer. And this has been actually a case in system neuroscience. People repeat an experiment, they see a different filter, and they just wonder why. Okay? And so now we have sort of, in this simple theoretical model, a clear explanation for this. Okay? Because we are saying, look, depending on the class of input signals you apply, you're going to get a different answer. And of course, what people do is they say, choose inputs which are sort of naturalistic signals. Okay? Naturalistic signals determine a class of filters. Okay? And depending on what class of filters you're picking, therefore, you're going to get a different answer. The results are very powerful. Why? Because basically what we can do now is, by transforming the system identification problem into a neural encoding problem, we can sort of talk about recovery of the projection of the filter precisely, and we can connect it to Sharon theory. Okay, so if the average number of spikes, let's consider this picture now first. So I'm looking at this branch only. Disregard the fact that this is a 1 here, so just think this is a U. So the input is P of H, then you have a filter U, and you have the integrating fire neuron. So the question now is, if you observe these spikes, what can you say about the input? In other words, can you invert this operator? This is linear and nonlinear cascade of operators, and you want to invert this. And this is possible, provided that the density of spikes here is above the Nyquist rate. So if you feel comfortable with information theory, then this becomes sort of an obvious statement. Of course, what's not obvious about this is the information is in the time domain, not in the amplitude domain. There's no clock involved here. So it's not Sharon sampling in the classical sense. It's in the sense of representing the information in the time domain There's no clock involved here and all that. But you can ask the question of inversion. However, this inversion is going to work only if the density of spikes here is larger than the Nyquist rate, which means that the bandwidth of the signal being encoded has to be represented here at the output by increasing the average number of spikes by a factor of 2. Intuitively, what that means is this. If your signal is 25 hertz, you essentially have to have spikes, which in average, they're running at 50 hertz. Mind you that this is an average statement, but actually what we need is the precise spiking behavior. Now, in system identification, however, what it can arise is that if you're just looking at this single branch, you don't have enough data here. Okay, if you don't have enough data, if you try to invert this operator, you're not going to be able to, or you get a very poor answer. But typically, the problem is ill-defined. So what do? You might want then to repeat the experiment with a different waveform, provided that these waveforms now, they are, let's say, independent, linear independent. Then you can amass enough data here at the output so that the whole thing can be inverted. So this is now a population encoding problem. But in the end, you see, all you have to do is just recover this P of H, and it doesn't really matter. So in other words, what you can do is this, practically speaking. You either repeat the experiment, and every time you record this data, and then in the end you invert it, or what you do is you run one single experiment where you run a waveform U1 for a while, then a waveform U2, then a waveform U3, so that in the end you have enough data. What does it mean that in average you have enough data? What it means is that if you you put everything on a column, okay, then the number of spikes in average is beyond the Nyquist rate. And then all this can be inverted. And here's the representation of this recovery. It requires inverting a matrix. There's a pseudo inverse involved here. It's very easy to implement. I'm not going to go into details on this. Suffices to say is that we have now this result which says that we can identify the projection of the filter on the input space with arbitrary precision. That has not been possible to talk about in the past in system identification in neuroscience. So if you look at LNP models, there is an approximation. There is some sort of a filter. It's not clear, so what sort of theoretical statements you make. But furthermore, you see we're working with sample path. And as a result, we run an experiment for a while. And from that, we can identify the projection of the filter with arbitrary precision. OK, so here's an example to give you a sense of this. So here's the input signal. It has 25 hertz. We are using a Hilbert space which has a reproducible kernel called the Dirichlet kernel. What it means is that we essentially have a finite number of spectral lines. In this case here, we have 5 spectral lines on 25 hertz, so we place them at 5, 10, 15, 20, 25 hertz. This is how we generate the signal. This is the input to the neuron. This is the input to the thresholding device, and then these are the spikes now. And now look at the recovery. In dashed line, you see the filter itself, and in red is the perfect recovery of the projection. But you see there's a difference between the projection and the actual filter. Okay? And you can see this in frequency domain. Essentially, what's happening is this, that if the filter has sort of large bandwidth, with 25 hertz, you cannot cover the bandwidth, and therefore you have this difference between the projection of the filter and the filter itself. Now, however, this can be further analyzed, and what we can do is now we can increase the bandwidth of the class of signals, test signals at the input to do system identification. And I'm showing an example here where we're picking 4 signals. The bandwidth is now 100 hertz. And you see now that the difference between the filter and the projection is really tiny. It's around 48 dB. That's sort of precision, roughly 8 bits precision. And so from a system neuroscience standpoint, that's more than enough. If you're looking at it as a maybe electric engineer, then 8 bits maybe is not enough, okay? But for the application here, it's more than enough. And of course, if you want to do better, you just increase the bandwidth. And that requires now that your experimental capabilities are more powerful in all this. From a theory standpoint, you see we are providing here a methodology where we can tell when to stop. Now, in terms of intuition, there are some examples here. If the signal is, let's say, 20 hertz at the input, 50 or 100, the actual filter is in red. The reason what you see here is there's periodicity because these are periodic signals we deal with. So the signal in the filter is in red and the approximation is in blue. And you see at 20 hertz there's a huge difference between the two. And you increase it to 50 hertz, that gets closer. And at 100 hertz you don't see a difference anymore between the two. And of course this can be then substantiated in the frequency domain. Essentially what happens is in red you see the Fourier transform of the filter, and so with a bandwidth of 20 hertz, this is the approximation you do. So it's a very good approximation in this band, but nothing outside of it. And then as soon as you start increasing the band, you get closer and closer. And of course, from a theory standpoint, you can approximate this as well as you want to. So the next step is to move to random thresholds. And that's related to a question you had. So random threshold essentially means a simple model here, integrate and fire with random threshold. So it's a threshold which, let's say, varies according to a Gaussian distribution. So between two consecutive spikes, you have a fixed value, but then in the next two consecutive spikes, the value is different. And then might be Gaussian, okay, for computational purpose. Setting this up, now it's slightly different. So again, what we want to do is, knowing U and knowing the time sequence T, we'd like to identify H. Now, the identification of H cannot be done anymore with perfect precision. So what one has to do is one sets the problem up using Tikhonov regularization. So there is a whole machinery for this. It's actually quite easy, quite simple, and then look what type of results we get. Here is an integrated amplifier with random threshold, 2 test signals, and you get the P of H is now in red. We use something like 26 spikes here, and the precision is around 30 dB. 30 dB means roughly 5 bits. Okay, so noise obviously has an impact on this. It's a fact of life, you cannot get around it, but in effect, you can recover this with high precision. And the formula is, again, I'm repeating this, but you see the formula is very powerful from the point of view that now we have a formal connection between neural encoding in terms of time encoding and system identification. So we write theorems about this. And now the issue is, can you actually extend this? And this is what I'm going to do now. So I'm going to assume now that I have multiple filters at the input, and I have a spike neural model. And so it's like intuitively I have a dendritic tree, and there's a lot of stimulation going on from multiple waveforms in parallel. For instance, in olfaction, that would mean maybe 2 odors simultaneously accessing the same cilia. And so we have the same problem here, except that now this is more interesting because we have a number of linear filters. It's not just one. Intuitively, the methodology is the same, because all what we did is we just increased the space of the filters, the linear operators. So instead of having a scalar linear operator, now is vector. But in effect, the same conceptual methodology applies. And so in the end, what that means is we can write down, again, the same formulas. And what we end up with is now is a neural encoding problem where we have multiple inputs. Okay, we have a population of neurons with random threshold encoding this vector input. Again, remind you, the vector input is due to the projections of each of these filters on the input space. And so the intuition here is, again, the same. You had the filters. You can work with the projection of the filters. You swap the projection with the input. So the input becomes actually a set of filters. And so we end up with a neural encoding problem, which we know how to deal with. And there's a precise formula how to do this. And so here is a set of examples. We are picking here 3 waveforms. I'm showing 3 waveforms in red, green, and blue. And so what we have to do is basically we have to find, we have to identify 3 filters. You see the problem here is like having 3 filters at the input. So that's exactly what we do here. We have 3 filters, and we identify the 3 filters. And again, you see we can come up with a pretty acceptable solution from the point of view of projections by using essentially a bandwidth at the input of something like 100 hertz. Okay? The conditions of the problems are similar to what I had before. I don't think I have to go into details, but you see now simultaneously we are getting 3 filters out at the output. So it's like encoding. You see, the issue is there's a single input, U, in the beginning, and you have 3 filters. And you have to identify 3 filters from a single input. And because there's not enough data, what you do is instead of having a single input, you repeat. You have a number of inputs, but the dimensionality of the input is still one. Okay? So 3 dimensions as far as the filters are concerned, but 1-dimensional at the input. And so these are the 3 filters which are then computed simultaneously. And to get enough data, then what we say is, put at the input multiple waveforms. So it's like the standard, methodology in systems neuroscience. Have an input, stimulus, get the data. But we are not computing the PSTH. We are looking only at the spike trains. You see, this is a set of spike trains here because it's a variable threshold sequence. You see, this is the average threshold, and then it's varying around this. And of course, you get a raster plot accordingly. So the issue is now that we have a quite simple extension of this, where we can essentially go to a multi-input, single-output environment, and we can identify the filters, of any dimension, basically. Okay. So what I have done until now was basically I was looking at the simplest models which are out there, integrating fire with random threshold. And there's a precise formulation of this. Now, what we'd like to do is we'd like to go to the class of conductance-based models, which neurophysiologists feel more comfortable with. Okay, mathematically, there are some differences, obviously, but one can handle this using essentially it's the same machinery. So now, what we're going to assume here is this: that we have a neuron which is described, let's say, in the Hodgkin-Huxley case with 4 differential equations. I presume I don't have to write them down. You have seen them many times. So we just write this like this. Hodgkin-Huxley dx is equal to fx dt. And x is x1, x2, x3, x4. So x1 is v, x2 is h, x3 is n, x4 is m. And so the 3 gating variables and the voltage equation. So this is a very nasty differential equation. Even if you're looking at it from purely mathematical standpoint, it's very difficult to deal with. And now what we're going to do is, in addition to having a stimulus input to this, so it's only, we are stimulating only the first equation, the voltage equation. In addition to this, we assume now that also we have noise. So the last 3 equations, the gating variables, have essentially winner process input. Okay, so there are diffusions, and so the question is how to deal with this. the mathematics is, as I mentioned before, can get out of hand pretty quickly. Nonetheless, we need to handle this in some manner. And so here is the description. I'm just taking the very simple case. I have two filters. It doesn't really matter. So I have two filters. The inputs are U1, U2. The outputs V1, V2. There's a bias added to. This is a description of the neuron. And the spikes are recovered through some zero crossing detector. For instance, one looks at the peaks of the spikes, or maybe some zero crossings. Okay, it doesn't really matter. We tend to write these formulas in this way because it applies, let's say, directly to neuroscience. And you see here Hodgkin-Huxley or any conductance-based model. But if you are coming from communication theory, this looks like a frequency modulator. Okay, and so there are some people who are very interested in that. And it turns out that the formula is actually, mathematically speaking, is the same. Mind you, frequency modulators work at 100 megahertz, okay, and neurons work at 100. In spite of that, the formula is the same. So it's quite neat from that standpoint. Okay, so this requires a machinery which is quite extensive, and I'm just going to give you some intuitive insights in this and how we go about this. Fundamentally, the problem is that if you have let's say, Hodgkin-Huxley neuron and it's tonically spiking, then you have a single limit cycle, basically. Okay, and what you see is something like this. Okay, but if you actually have an input to the Hodgkin-Huxley neuron, then essentially the limit cycles sort of vary. So here I'm representing basically limit cycles in 2 dimensions. We are looking here at the one of the gating variables and the voltage. And, this is a 2-dimensional space. And if you apply an input, like it's shown here, blue or red, essentially you start wandering around this here, and you're never on the same limit cycle. So mathematically, this object is very difficult to deal with, bifurcation theory notwithstanding. You cannot apply bifurcation theory to this, okay? Because, essentially you have a function of time as an input, and the system has a much more complex behavior. Now, in red, we show exactly the times when the spiking occurs. Okay, so you apply an input, and so essentially then spiking occurs around these times. Blue comes from the fact, from the, let's say, the blue curve here, and red from the red. And so you could say the following, that if this red plot here, meaning the red input, is weak, then more or less you're on a single limit cycle, sort of stable limit cycle. Otherwise, if it's blue, sort of you wander around. And so this is highly nonlinear. And the question is now, how do you go about this? And I decided not to go into the details of this, but just to give you some sort of a sense, at least for those of you who are sort of mathematically interested in this. We work with the phase response curve. Actually, we work with an extension of it. It's called the conditional phase response curve, which we sort of devised for this purpose. And what it does is, essentially, it estimates the limit cycle the system is on in real time. So just by looking at the spike sequence, you can roughly know what limit cycle the system operates on. And one can use that information in order to reduce the entire system to something simpler. Still non-linear, but something simpler so that one can actually deal with it, okay? And that's called, one ends up with something we call a project integrating fire neuron. It sort of looks similar to an integrating fire neuron, but the phase response curve comes in there from here, from the Hodgkin-Huxley neuron. And if you do this, then here you can see the recovery of the filters. And the system here, we assume that is noisy, meaning that it's only the Hodgkin-Huxley neuron, and the noise applied through the gating variables is 0, okay? And as a result, you see this phase response curves, which are pretty smooth, okay? Now, if you want, of course, to see the next case is we start applying some noise to the system, and then you can see that now this sample path, they start wiggling around. So this becomes more difficult to treat from a mathematical standpoint, but if you look at it intuitively, it says, well, roughly speaking, one should be able to deal with this. If we dealt with the previous problem, one should be able to deal with this too. And that's the case here. And you can see, the recovery of the filters, okay? Again, the precision here is around 23 dB. And if we increase now the noise value, some higher number, the actual number is not so important. You see the system becomes even more wiggled. And so this additional noise leads to some drop in terms of recovery. But again, we can recover the two filters which stand there for modeling the input dendritic tree. Finally, this can be sort of analyzed in terms of how many number of spikes one needs so that one comes up with a fairly efficient way to recover the system. And you can see sort of if the number of spikes is high enough, then one comes to some sort of a steady state situation. And here are essentially the filters and the projections represented for the various cases of the signal. So it's not so important at this point, the details. I think the important fact here is the big picture, which is that if we add noise to the system, we have a fairly good handle of what sort of performance the system is going to exert on this recovery. And that's the critical aspect of this. So until now, what I have done is I have looked at one and multidimensional filters at the input, but they were all temporal filters. So the assumption was I have a neuron, okay, as a dendritic tree, there's a stimulus applied to this. The tree appears as a linear operator in the picture, but everything is in the time domain. So it's a classical time filter. So now what I'm going to do is I'm sort of extending or generalizing these results to the case where we're dealing with a spatial temporal receptive field. And so here's an example. Here's a Gabor filter depicted in 3D. So there's an XY coordinate here, and this is the value of the filter as a function of x and y. So it's f of x and y. And there is no time component in this. So the filter is a pure spatial filter. And now the issue is identify this. Okay, now of course the problem is more interesting conceptually, but also computationally, because you start actually getting sort of some screen sizes here of 800 by 800 pixels. And so the way to think about this is basically that you have a filter like this, a spatial filter, and this is in front of the neuron. And essentially this filter acts on a set of images, okay? And the result of this filtering is going to drive the neuron. And then you observe the one-dimensional output. So again, the input is multidimensional. The output is a time sequence. And you would like to actually recover this. And here's the recovery. One example. You see you can see the absolute error here. Essentially, it's tiny. And you can see that using contour plots for the original, for the recovered, and for the difference between the two filters, and the same thing in the Fourier domain, okay, in the spectral domain. So what we have here is the situation where we extended the problem to a spatial dimension, but of course, what one would like to do is have the same capability also for the non-separable case. In other words, now the input filter is not just a function of x and y, but it's also a function of t. And of course, if the two are separable, then you can deal with the two problems separately. Okay, so you identify one, you identify the other, and essentially you can solve it the way we discussed it before. But what I'm showing here is a more interesting case. And the case is where the receptive field is non-separable. And non-separable receptive fields means now that the filter is a function of x, y, and t, and you cannot write it as a function of x and y multiplied with a function of time. So here's an example of this. Now note that there's a time sequence here, so 10 milliseconds, 20, 30, 40, and the shape of the filter changes. And we are using here a rotation, so you see the filter is actually rotated. So the xy direction, there's a rotation happening, and it's a function of time, and so the receptive fields are changing as a function of time. And so this is the filter itself. And now we are showing here the projection of this filter. In other words, what we are recovering through our methodology. And here is the result, and you can see the precision. We worked this for high precision. It's like 100 dB. 100 dB means 16 bits precision. So it's like audio CD quality, okay? At least vintage, I don't know, 10 years ago or something, okay? So it's extremely high quality, and you can get this very precisely. And of course, you can do this then with other operations, and you can see I'm looking now at translations, and you would guess that the next slide is going to be about scaling. These are sort of group operations, so that are very interesting, especially for image recognition. I can say that there are quite some experts here in image processing here. So this is of interest because in image recognition, basically, we recognize objects independently of scale, translation, or rotations. Okay, and somehow these operations have to be then understood in the spike domain. And what we're doing here is essentially looking at filters which have these properties and see how we can deduce this. So here are the dilations, the same thing. You see this is very small, and here this is very large. So picture is dilated. So maybe I should show a video. So here is a dilation. You see the Fourier transform is sort of changing. Now we're going to a different filter. We just changed one of the parameters in the Gabor filter just to exemplify it. So you see, basically, this is the difference between the two. Now we have rotations. In the case of rotations, it's easier to see what's going on. You see the rotation happening there in the time domain, and here rotation in the spectral domain. For various examples of parameters of the Gabor filter. Then here are translations. You see the filter essentially translates from left to right. The spectrum doesn't, of course. You don't see that. But in terms of identifying the filters, we can do it with extremely high precision. And so at this point, you see the progression has been, we started with temporal filters, and we said essentially we would like to identify a filter H and for any spiky neural model. And we went through integer-unifier, Hodgkin-Huxley, any conductance-based models. Then we said, okay, now how about we increase the number of filters on the input? So the input is sort of a vector of filters, if you like. And now this is not a vector anymore, it's a spatial filter. Okay, in many ways, this is very interesting for different type of applications. So the vector case is interesting for olfaction. The case of space-time filters, of course, is interesting for video. It's interesting for audio. People in both of these fields, apply these filters, and so you would like to identify those. But every time when we did this, we had an input which was basically a continuous waveform. And as soon as you move away from periphery, as soon as you move away from the retina, cochlea, or olfactory sensory neurons, basically what you're going to encounter is that each neuron has an input. It's a train of spikes, and the output is a train of spikes. And now what you have to do is you have to do a system identification problem for a train of spikes, okay, input and train of spike output. And that problem is, of course, very interesting. Also, if you do, let's say, work in vitro, you want to apply some train of spikes to your neurons, and you want to see what the behavior is, and from there, can you actually deduce what the neurons are doing? And so this is the last topic I'm going to present here. So I'm looking at this model now. I have a neuron here which receives spiking inputs from 3 other neurons, as an example. Of course, the dimensionality doesn't matter. And so the notation is the input is S, K, 1, 2, what have you, and then we have a spiking neuron here. And so the issue is spiking input here, spiking output there. What can I say about the input? Okay, and again, I'm not going to go into the methodology, but I'm just giving you an example of how this works. So here I have the train of spikes in 3 colors because I have 3 inputs, and each of the inputs actually acts on a filter. So remember, I have a dendritic current, which is the aggregate dendritic current coming from here. You see, I have these 3 inputs, and I have the 3 filters, and the result actually is summed up and drives actually the axon hillock. And so this is the input, and here we're showing the aggregate current, which drives the axon hillock, or the spiking neural model. And here's the spike train at the output. So there are no colors because this is, of course, a single spike train. Okay, so the multiple spike trains are the input, multiple spike trains are the output. Now, why is this problem interesting? From assistive neuroscience standpoint, it's clear. This is the essence of anything which is going on. Most of the neurons are not at the periphery, so it's exceedingly important. From a theory standpoint, this is interesting too because here you have spatial distributions. They're all delta pulses, and here there are delta pulses too. And you have to essentially work with 2 spaces, which they have their own intricacies. And so the question is, how can you essentially come up with a framework, mathematical framework to deal with this? And now, so we again apply similar ideas to what I described, but I'm not going to go in detail about it. However, I would be happy to discuss this, later on. And here we do the identification of the 3 filters, and you see again, essentially we get extremely high precision of the 3 filters, simultaneous identification for a single output sample path. So if you like, if you really want to do this, in principle, you can do this in real time. Okay. So let me stop here. Not before just concluding. We have made a connection between system identification and neural encoding. The neural encoding question, some of you might recall I discussed this 3 years ago here in the context of time encoding machines and time decoding machines. And that field now is at a point where you can take an HD quality video, 640 by 360, let's say, number of pixels, and you can look at the recovery. Essentially, there's no difference between the original video and the recovered one. And we can also do operations in the spike domain, directly in the spike domain, operations such as translations, rotations, and scaling. In other words, an image comes in, you have a encoder, and you can then from the spike stream, you can do a manipulation such that a recovery, what you see is a translated, scaled, or rotated version of the original. So that body of theory is in the classical sense of what you see in information theory, what you see in communication theory, where you have an encoder, which you have some observations, and you want to say something about the signal which has been encoded. Here the problem, and that leads to a inverse problem, classical inverse problem if you like. Know the box, know the output, meaning know the transformation, know the output, but you don't know the input. In system identification, the problem is in some ways similar, being also a inverse problem where you know the input, you know the output, but you don't know the box. And what we have done is we have connected the two. And by connecting the two, of course, we could essentially then move from system identification to time encoding, neural encoding, in a very transparent way, with statements, theorem, proof type of statements. Now, from a system neuroscience standpoint, the results are important in the following sense. There are a class of identification problems where you cannot apply white noise analysis. As I showed you in olfaction, is not possible, number one. Number two is the methodology does not call for repeating the experiment in the sense of computing the PSTH. This is single path. This is language from, I guess, stochastic processes. You have to do a single experiment, okay, to recover the input. And obviously that it's a great help because you don't have to repeat the experiment. So instead of sitting 6 hours there and hoping that your fly is still alive, you can sit there for half an hour and you're done with it. So it's really a very practical big deal. Okay. So that is from our standpoint extremely helpful. And of course the machinery can be applied now for essentially every known neural model out there, conductance-based model, if you prefer to work with integrated fire, threshold and fire, what have you, it all fits into this context. So with this, thank you very much.